Today, we are going to be learning about a research article titled Rapid and Reversible Control of Human Metabolism by Individual Sleep States, which makes key connections between the processes the body takes to make and break bonds and the effects of sleep surrounding that. In order to understand the paper, we have to know key definitions relating to metabolism. Now, you may be asking, well, what is metabolism? Metabolism is the entire set of enzyme-catalyzed transformations of organic molecules in living cells. There are actually two parts of metabolism, which are catabolism and anabolism. The figure shown on the right is found on page 27 of the book and is a really good example of metabolism, showing the central roles of ATP and NADPH in metabolism and how they are utilized for catabolism, which is the breakdown of metabolites, and anabolism, which is the synthesis of simpler molecules for energy. But you may be asking, well, what are metabolites? Metabolites are small molecules that are the intermediates, which happen after catabolism, or the end products, which happen after anabolism in metabolism. These metabolites make up the metabolome, which is the complete set of metabolites in a biological system at any given time. The study of the metabolome and its functions and reactions is called metabolomics and is exactly what today's paper explores. If the paper is talking about metabolism and sleep, where does sleeping come in? Well, when you are awake, a molecule known as glycogen, which you can see here on this picture, is broken down into a usable form of energy storage called glucose, allowing you to use energy for metabolic processes, going down through glycogenolysis and all of these other metabolic pathways. Glycogen molecules are then synthesized and regenerated when you are asleep through glycogenesis, shown here. Well, why is this even important? Well, systematic disruption of sleep and wake patterns, aka your natural circadian rhythm, results in considerable disruptions to normal metabolic patterns like those we just mentioned with glycogen. These disruptions are believed to link together shift work and metabolic syndrome, which is a wide spectrum of disorders. These metabolic syndromes include obesity, diabetes, and cardiovascular disease. Each of these diseases is a leading cause of death globally. In 2021, with just the United States, around 300,000 people died from obesity-related complications. Around 400,000 people died from diabetes-related complications, and around 695,000 people died from cardiovascular disease. Today's paper takes initiative into figuring out how normal human metabolism is affected by sleep cycle disruptions and just how important sleep is to these processes. To determine the relationship between metabolism and sleep-wake cycles, these researchers measured exhaled breaths in 10-second intervals of 13 healthy individuals with normal sleep patterns. The apparatus shown in this picture, figure 1A from the paper, is how they non-invasively collected these exhaled breaths. Molecules from the exhaled breaths were ionized, and those ions were detected with a high-resolution mass spectrometer. A full polysomnography was performed in parallel with this, which measured the test subjects' movements throughout the night. Mammals, like humans, have their sleep divided into two different categories: REM, or rapid eye movement sleep, and non-REM sleep. Non-REM sleep falls into three further stages, which are N1, N2, and N3 sleep. N1 and N2 sleep are the stages changing from wakefulness to deep sleep, which is N3 sleep. As you can see here, this is a graph of different metabolite levels collected with a mass spec of 3 different individuals measured using polysomnography. You can see here how the direct increase in metabolite levels increase with REM sleep. Remember earlier when we said that during sleep the body regenerates and synthesizes glycogen? Well, during that time, not only is glycogen synthesized, but so are 2,000 other metabolites. As it turns out, lipid metabolism, carbohydrate metabolism, and the TCA cycle are all sleep-wake state dependent. Let's look into the carbohydrate metabolism and see what happens during sleep. Let's go step by step into important findings from the paper and what pathways those would be in. When examining glycolysis, the researchers in this paper found the highest glucose levels during wakefulness and the lowest levels during REM sleep. This correlates with an increased glucose utilization during glycolysis for REM sleep and a decrease in glucose utilization during non-REM sleep. Well, what does that even mean? Well, glycolysis is the pathway that breaks down glucose into pyruvate so that it can enter the citric acid cycle for cellular respiration. As you can see, after just one round of glycolysis, you only have a net of 2 ATP molecules, which is not enough for any other metabolic processes or body functions. Some of these processes, like cellular respiration and biosynthesis, require a lot more ATP and or constant ATP, and these processes need ATP and starting reactants to function, meaning a lot more than one round of glycolysis needs to be performed. Look at all of the other processes that glucose is involved in. All of the processes shown need glucose at some point, and there isn't an infinite supply of it. Your body has to convert glucose from food, and if you aren't eating, your body needs another way of continuing to replenish the glucose you use for your metabolic processes. That's where glycogen re-enters the picture. Let's take a look at how glycogen is synthesized so that when you are not using REM sleep, your body can regenerate this glycogen using a process called glycogenesis. For glycogenesis, glucose needs to be taken up by the liver or muscle cell from the bloodstream when you eat. Once that is inside a cell, glucose is converted into glucose 6-phosphate by an enzyme called hexokinase. This conversion keeps glucose in the cell so that we can further metabolize it. Glucose 6-phosphate is then converted into glucose 1-phosphate by an enzyme called phosphoglucomutase. That glucose 1-phosphate is activated by the addition of a molecule of uridine diphosphate, UDP, and that forms UDP glucose. This reaction is catalyzed by the enzyme UDP glucose pyrophosphorylase. After that, a small glycogen primer molecule is formed by attaching several glucose molecules to a protein called glycogenin. There needs to be at least 4 of these molecules attached for this process to begin. That primer will serve as the starting point for glycogen synthesis. UDP glucose molecules are added one by one to the growing glycogen chain, catalyzed by the enzyme glycogen synthase. These are connected using alpha-1,4 glycosidic bonds. This process occurs with the assistance of branching enzymes, which create branching points in the glycogen chain to allow for more efficient storage. Those branching points connect the glycogen molecules using alpha-1,6 glycosidic bonds. After the glycogen chain elongates, branching enzymes create branches by transferring segments of the chain to different locations, creating a highly branched glycogen structure. If glycogen synthesis can't be performed, your body doesn't have a way to store excess glucose. Today's paper found that sleep loss has been associated with impaired glycogen synthesis and glucose metabolism. This sleep loss decreases insulin sensitivity, and glucose can't efficiently enter cells where it can be used for energy or, in this case, storage. When your insulin sensitivity is decreased, that's called insulin resistance, which is closely linked to diseases like type 2 diabetes and obesity. All of this over an impaired and disrupted sleep-wake cycle. Okay, let's recap what we learned. We learned that glucose is needed for energy and is either used or stored in the body as glycogen. During non-REM sleep, your body uses less glucose, meaning less glycogen is being used. Glycogen and other metabolites can be regenerated and synthesized during non-REM sleep. Having a consistent and non-impaired sleep-wake cycle can decrease your chances for insulin resistance that leads to metabolic disease. For your first quiz question, in your own words, how is a regulated and non-impaired sleep-wake cycle important for your metabolism? Be specific and give an example of what would happen if someone had an impaired sleep-wake cycle. Here's your next quiz question. There are two options for this. I want you to find another research paper that either explores impaired glycogen synthesis or another paper that ties diabetes to a poor sleep-wake cycle. If you choose the first option, the impaired glycogen synthesis paper, what scenario was researched and what was the paper's conclusion about the impaired glycogen synthesis? If you picked the second option, the paper about diabetes and a poor sleep-wake cycle, did the paper make any other conclusions, and were any other metabolic pathways mentioned? I look forward to hearing your responses, and I hope you enjoyed this presentation. Thank you!